Hello, my name is Yugjeet Grewal, and I'm a junior at Fresno State. I'm also part of Dr. Blumenshine's Aquatic Ecology Lab, and today I will be presenting my work on growth of juvenile Chinook salmon and transition from yolk to environmental carbon and nitrogen. Let's talk a little bit about the history of these fish in the San Joaquin River. As you might know, Chinook salmon are anadromous fish, which means after their eggs hatch, they stay in the river for a few months to a year, and then they travel downstream to the ocean where they grow and get ready to reproduce. Then they travel upstream to their native stream, and that's where they reproduce. Once upon a time, San Joaquin River had one of the largest spring-run Chinook salmon population. This population consisted of hundreds of thousands of individuals returning every year to reproduce. However, in 1942, Friant Dam construction was completed and the river began to dry. With that, the fish, they weren't able to make their way back to the river to reproduce. And for over 60 years, we saw no Chinook in the San Joaquin River. On the bottom right, you see a picture of the Friant Dam, and on the bottom left is a graphic where one of the advocacy groups termed the San Joaquin River as one of the most endangered rivers in America. San Joaquin River Restoration Program began in 2006 after years and years of litigation. It aims to restore water flows that can maintain a self-sustaining and self-reproducing Chinook salmon population. The biggest challenge that this program faces is reestablishing a population that disappeared decades ago. Therefore, it is really important that we study their interactions with their environment. And that's where the focus of Blumenshine Lab lies. You might ask, how do we study their interactions with their environment? One of the answers is using stable isotope analysis. This analysis is done using mass spectrometry, and it measures the ratio of heavier stable isotopes such as C13 to the abundant isotope using standard as a reference. This is a way to denote stable isotope analysis, and it can be read as delta C13. Something else we know is that consumers' carbon and nitrogen signals, they resemble that of their prey. This is due to isotopic fractionation, which states that the consumers, they have carbon and nitrogen signals that are a little bit higher than those of their prey. And this concept is called enrichment. So basically, the carbon signal increases by 1 per mil and nitrogen by 3.4 per mil on average. We can use this relationship between the prey and the consumer to study trophic interactions using stable isotope mixing models. What these models do is that they basically match the consumer to their prey, and they basically tell us, for example, this taxa might count, one taxa constitutes 20% of their diet, and another one constitutes 30% of their diet, and so on. This graphic just shows how carbon and nitrogen go from primary producers to consumers, primary and secondary, and then to fish. And when we work with these stable isotope mixing models, we basically start with the fish signatures and the prey signatures, and the models basically match the fish to their prey. My focus is to study the change in carbon and nitrogen over time to see how it's changing. And it basically depicts how their diet is changing over time. My methodology is to dissect Chinook salmon dorsal muscle tissue samples, which were obtained from 82 different individuals. They were dried, encapsulated in tin capsules, and sent to UC Davis for dual carbon and nitrogen stable isotope analysis. We did similar processing for invertebrates as well. On the right, you see a picture of one of our smallest fish along with a bigger fish. And this is a tissue dissected from this particular individual before drying. And these are some dried invertebrates. Here are the results that we obtained. So this is a graph. The one on the left is a graph of delta C13 versus weight. And on the right is delta N15 versus weight. Orange triangles are from an upriver site, and blue circles are from a downriver site. As we go along x-axis, the size of the individuals is increasing. So what we saw is that smaller fish, they have abnormally high carbon and nitrogen signals. And these signals, they decrease rapidly as the individuals increase in size, showing a potential relationship here. And the same thing happens for delta N15 as well. We observed the same trend for our 2019 and 2020 fish as well, where we had some smaller individuals with very high carbon and nitrogen signals and some bigger individuals with lower carbon and nitrogen signals. Something I must note is that all of these individuals across both years came from the same site upriver. And for 2019, we did not have many bigger individuals. So then why do we care about these abnormally high carbon and nitrogen signals? Because these abnormally high signals can produce inaccuracies in stable isotope mixing models. These graphs are called mixing polygons, and they serve as a precursor to test the samples for if they would be a good cohort to run through the stable isotope mixing models. So these basically plot the nitrogen signal versus carbon signal, and the crosses are the prey or the sources, and the dots are our fish or the consumers. The outermost line is called the 5% contour, and the farther a consumer falls from this red area, the lower the probability that its carbon and nitrogen signals can be explained using the sources that we have. As you can see, the graph on the left only includes the fish above 3 grams, and they all fall nicely within this 5% contour. And therefore, we can use this data for stable isotope mixing models. However, if we include all fish, including the smaller ones, these fish fall very much outside of the 5% contour, and they shouldn't be used for mixing models. And these mixing polygons were made by Michelle, who is a graduate student in our lab. So we saw that the larger fish, they match the expectations based on isotopic fractionation, which means that they fall nicely within that 5% contour that we just saw, but the smaller fish deviate. And something that's significant here is that these smaller fish are around 0.3 grams, which is the stage where they have just absorbed their yolk sac that they obtained from their mother, and they've just started to feed exogenously. So we believe that it is due to the effects of maternally derived carbon and nitrogen that their signals are so abnormal, and these effects will fade away as they feed more exogenously. My current work focuses on working with fish that are between 0.4 to 5 grams, because in the previous years, we either had very small fish or we had some bigger fish that were above 5 grams. So I want to see what's going on in this particular area between 0.4 grams and 5 grams. And I'm currently processing 2021 and 2022 Chinook salmon. And the good news is that some of the individuals that we have already obtained are between this particular weight range that we want to focus on. And we are also trying to obtain hatchery raised kokanee. And the benefit of these is that they have been fed on the same diet. So they remove a potential confounding factor that applies to our Chinook salmon obtained from the river. So in the river, the stable isotope signature changes temporally and spatially with changes in prey concentration and taxa. But this does not apply to the kokanee. So we can remove a potential confounding factor and get a stable isotope signature of the diet and easily calculate exogenous incorporation rates with using these fish. And then perhaps we can see at what point have the fish incorporated the river prey base stable isotope signature and compare our results with the mixing polygons. Here are my references, and thank you.